We are live. Great. Welcome, everybody. Welcome to iNAV 2020, the third Interdisciplinary Navigation Symposium. As you can see, there's only me and Jeff Taubie online. But of course, there's more there's more than the two of us. This has just to do with technical limitations of Crowdcast, which doesn't allow a certain number of video streams. So next slide, Josh. Yeah, so beyond myself and Josh, there we go. Yeah, so we also, of course, have Laurent Direct from Paris, who's organizing this. Jeff from Dartmouth, myself from Magdeburg, Germany. Josh Jacobs, who's on here, but whose video is not live yet, from Colombia. Then we have Sylvia Wirth from Lyon and Mayank Mehta from UCLA. Next slide. Yeah, as probably most of you know, the plan for iNAV 2020 was a bit different in that we were supposed to meet in this beautiful location here in northern Italy in the Dolomites, in the town of Cortina d'Ampezzo, and everything was all set with a beautiful hotel. We already had made plans for outings on Wednesdays. And in addition to that, of course, as usual in iNAV, the meeting was timed perfectly to happen in parallel due to major football tournament, or soccer for North America. Yeah, unfortunately, due to COVID-19, we had to cancel this and change our plans. And instead, we got this. So, Josh, do you want to advance? Yeah, so instead of being in this beautiful location, now we're all sitting at home trying to do home office. And yeah, I have to say, though, it's on the one hand, it's sad. On the other hand, though, I think there's a big advantage. So when I look at the number of people who've registered, just as we speak, the number is at 365. So I mean, this is actually probably double the amount of people compared to the last iNAV we had in Mont Tremblant in Canada. So it's great to see that many more people can actually join us here, and we're very excited to share the latest navigation research with you over the next couple of days. So I'm handing over now. Oh no, sorry, just one more slide to get before we go. So yeah, this is kind of what it looks, I guess, for many of us. But there are some people who are a little bit more privileged than us. Like, for example, Jeff Taubie, who sent me these photos yesterday and said this is what it looks like now from in his backyard. Beautiful Indian summer. So I'm gonna hand over now to a very relaxed Jeff Taubie, who's gonna tell you a little bit about how the whole thing will work out at Crowdcast and what to expect in the coming days. Thank you and see you soon.
Okay, hello everybody. I was just, before we went on screen, I was just telling everybody, I think I'm more nervous, or I'm as nervous now as I was when I did my PhD defense. This seems like it's like regular stage production. But be that as it may, here we go. Josh, can you bring up the next slide? So we have a great program planned over the next 3 days. The program is posted on the inavsymposium.com/program. So you can just go to that website, and the entire program, both for the regular sessions and the Blitz sessions, are up there. We're going to have 20 speakers over 5 sessions, 2 sessions today, 1 session tomorrow, and then 2 again on Wednesday. In addition, we're going to have 30 Blitz talks over 2 sessions. We had to add another session from that we originally had planned. So 1 Blitz session will be tomorrow, and then another one, a second one, will be at the very end on Wednesday to go over the regular sessions a little bit. We received many more abstracts than we can accommodate. We received about 70 abstracts, and so we had to cull that number down to something that would be a little bit more amenable to do Blitz talks in. So half of the people will be going tomorrow, and then half will be going on Wednesday. But nonetheless, we're going to publish all the abstracts that people submitted. So all 70 abstracts will be published in a little booklet that we will send out to everybody at the end of the conference. First of all, I must say sorry to everyone who might be in some inconvenient time zones. We realize that this might be a little bit, let's say, Europe and North American centric in terms of times, but it was the best that we could do to make sure we can accommodate the most number of people. So our apologies for those who might be in Australia, New Zealand, Asia, or elsewhere. And then finally, I want to give a big shout out. Whoops. Josh, hold it. I want to give a big shout out to Suzanne Wilbers, who's Thomas's sister, who has done a lot of the organizational work behind the scenes here. So my applause to Suzanne. Okay, next slide. So the way this is going to work is the whole conference, as you know, is going to be done with this application called Crowdcast. We're new to this too, just as you are. So you need to sort of bear with us as we go through in case there's any technical glitches. We did go through some dress rehearsals last week, so hopefully things will be okay. And the way this will work is I'm going to be the moderator for the first session, and I'll introduce the speakers. And then as the speakers are presenting their talks, if you have a question, there should be a little on your bottom line at the bottom of your screen, there should be a ask a question. And you just sort of type in your question there. And throughout the speaker's talk, there's going to be a poll that you can sort of take and say, if there's like 3 or 4 questions that appear, you can say which question is the one that you would most like me to ask the speaker at the end. So as we go through the talk, you should sort of answer that polls. And again, I will tally the counts at the end of the talk. So make sure you do that as we go through the talks, okay? And again, I will ask all the questions, at least for this first session, and the speaker will respond. So again, just as a reminder, make sure you ask a question and view the poll and put the question, vote for the question that you want me to ask. Okay. All right, I think this is the final slide. Next one. So just, this is sort of the slide with the big question mark, given where the pandemic is, et cetera. Just as a preview, what we're thinking is the next iNAV will probably occur in 2022. We'll sort of skip next year, and we'll be back on our even year schedule. Where it's going to be, we don't know. We might try again for Cortina in Italy in the Dolomites, but we'll see as we go along through 2021. But that's the plan right now. Okay, Thomas, you want to say any final words before I introduce the first speaker?
No, I think you did an excellent job. So
okay.
Looking good. So now we're going to have actually the possibility to see how time zone affects those unfortunate speakers from Australia, because we're going to have Michael Milford there. So I'll leave the green room now and leave you two on stage. So please, admin, kick me out, and I'll see you soon. Bye-bye.
Okay, so our first session is titled Circuits and Orientation. And we're going to have 4 speakers: Michael Milford, Stanley Heinze, Mark Brandon, and Alessandro Treves. And our first speaker will be Michael Milford from the Queensland University of Technology in Brisbane, Australia. I can't fathom what time it is in Australia right now, but Michael, I'm sure, will tell us in a minute. And anyways, the title of his talk is a long one. It's going to take me almost 30 seconds to get it out. Taking Off: Robotically Motivated Investigations into Bio-Inspired Mapping and Localization Across Species and Operational Domains. Michael.
Hello, can you hear me okay?
Yes.
Fantastic. Well, good morning, good afternoon, or good night, wherever you may be. Thank you so much to the organizers for making this happen, despite all the technical challenges faced. I'm looking forward to seeing all the sessions, as many as I can, and hopefully my kids won't wake up as it's 1 a.m. here and I'm just in the next room. So today I'd like to talk about a few things just to get us started. I realize the first speaker has a lot of pressure to explain grid cells and all sorts of concepts, and because I only have 20 minutes, I'm not going to try and do too much of that. I'm just going to present you with a few sort of things that we've been doing in our lab across biologically inspired navigation, and then leave everyone with hopefully some questions that other speakers can add to as we go throughout the conference. So just as context, I'm a bit of an unusual speaker, I think, at one of these events. I come from a very large robotics group at the Queensland University of Technology in Brisbane, Australia. And basically what we do is we take abstract theories of navigation as well as machine learning methods for navigation and put them on robotic platforms operating in pretty much every domain on Earth. So it could be a drone flying in the sky, it could be a robot working on a farm or in a mine site, or even a submarine robot working deep below the surface of the sea. And we encounter a lot of very interesting navigational and perception challenges in trying to do this. If you want more information, look us up online. I won't have time to cover anything in detail, but we have lots of survey and tutorial papers on a lot of the topics I'll talk about, and open source code of varying quality. Some of it's sort of what you'd call research grade code, and some of it is a little bit more polished and widely used. So just as context, I'm not going to talk about this really today. We've spent collectively nearly 2 decades working on biologically inspired robot mapping and navigation. One of our most well-known systems is called RatSLAM. SLAM is an acronym for simultaneous localization and mapping, and RAT means that it was inspired by rats. So all of the amazing navigational work in the mouse or rat brain that's occurred over the last few decades. And I don't have time to review all of this, but I guess we know that many of the key researchers in this field at this conference and have influenced this conference, and it's been a great source of inspiration for us. And just in one slide, RatSLAM is a continuous attractor neural network model of some of the navigational systems that neuroscientists think exist in the brain, but it also has some engineering scaffolding attached on. So in the middle of this diagram, for example, we have a graphical map readout of the encoding of space in this model. And this graphical readout provides us with a sort of more interpretable map of space, for which we use for many purposes. And with this system, we were able to do things like create maps of an entire suburb using just visual input from a camera mounted on a car. This was back before autonomous vehicle research was really a hot thing and had tens of billions of dollars invested in it. And there have been many other biologically inspired SLAM systems since. Most of the animals are gradually being knocked off one by one. And I think going forward, this is a great source of sort of inspiration for creating navigational systems for robots. So 3 pieces of work that we've been working on more recently. The first is taking some of the broad concepts of biologically inspired maps, especially these sort of multi-scale maps that are found in the mammalian brain, and applying it to a range of robotic applications, including moving into the sort of more aerial robotics domain. So grid cells, everyone knows about grid cells. I am not going to badly explain them in a rushed way, but basically an amazing encoding of space in the brain. So one of the things we've been doing a lot of, this is recent work with Boston University, is taking some of these concepts of encoding the world at multiple homogeneous scales, in this case in an aerial navigation context. So imagine a drone or perhaps an animal traveling long distances through our skies, and looking at how the use of a multi-scale map can aid in navigation performance. And there's a paper here with much more detail, but essentially using some very large data sets of traverses across large expanses of the environment are able to show that these multi-scale representations offer very significant performance benefits in terms of navigation, in terms of keeping track of where the robot or perhaps an animal is within the environment accurately and reliably. And I guess from a lot of this work that we've done over the years, we always come back to these key properties of these spatial cells. So I've rated these with asterisk properties of how important from a functional perspective we believe these are. So multiple scales, incredibly important. The exact scale ratio between the different map scales encoded, not too important. Setting the scales, moderately important. and how these different map scales may integrate sensory modalities of all types. So not just vision, not just olfaction, not just auditory, but all of them together, extremely important. We still can't find a compelling reason for the hexagonal or triangular tessellating pattern. I know there's been a lot of great theoretical work in this field, however. The memory and computational advantages are probably the most important property that we've seen. And we've used these for all sorts of nice little tricks in terms of robotic mapping and navigation. And the other overarching observation we have is that in cells like the grid cells, of course, we have varying levels of conjunctive properties, I guess, cells that have both positional and orientational information. We found that this conjunctive encoding is extremely powerful in terms of encoding and dealing with perceptually aliased or ambiguous perceptual data about where you are in the world, but that this encoding is also very expensive. So you need a lot of neurons in order to efficiently encode where you are in the world. So this is work that was led by Fangwen Yu, who visited with me for 2 years. I think he's on the call now, so hopefully I get his work correctly represented. And he's the founder of neuroslam.net. So I had a professor many years ago lecture me for about 3 hours at a conference that RatSLAM had negative connotations because of the word rat in it. So we fixed that by going to neuroslam, which has hopefully more positive connotations. And what we're really looking at here is how we can move to encoding 6 degrees of freedom of motion in a navigation context. So when you move through 3D space, you have, I guess, what's generally regarded as 6 degrees of freedom. So you have your XYZ position, and your vehicle or plane or animal can roll, pitch, and yaw as well. So altogether, that's 6 degrees of freedom in your movement. Now, you can take the continuous attractor models that we've been working with and many before us have worked with over the decades, and you can scale them up to an arbitrary number of dimensions, but it's not that simple when you're trying to encode 6 degrees of freedom motion. So this is an example of us thinking through how you can encode location and orientation when you move to 6 degrees of freedom. So you could have something like 3-dimensional place cells, and I know that some of the other speakers will actually talk about rat experiments and perhaps bat experiments in 3 dimensions later on. And the basic idea is that when your robot arrives at a particular XYZ spatial location, the corresponding cells in your network fire or activate to represent that you are at that location. When you try and integrate it with a head direction model that expands to, I guess, a 6 degree of freedom representation, it gets much, much messier. And Fangwen and I spent a lot of time over this 2-year period sort of working through some possibilities here. And these are not all of the possibilities, these are just some of them. So if you just need to represent 4 degrees of freedom, so yaw and altitude in a head direction model, you can have something like a stacked ring attractor model. So each individual ring in this diagram represents yaw or sort of orientation in a ground plane. And then you have multiple stacks of this to represent your different hypotheses about yaw at different operating altitudes. That's one model. You can go to a torus model. So toruses have been used in past place cell models as well. In this case, you can represent yaw and pitch concurrently in a head direction model, but you don't have roll encoding. So it's only 2 of the degrees of orientation that you're representing in this case. You can split it up into 3 different ring attractor models, so 1 each for yaw, pitch, and roll. This is very efficient in terms of the number of neurons you need, but you have a coupling issue in that you can't jointly encode combinations of yaw, pitch, and roll because these are all separate networks. And this was the problem that we encountered many years ago when we were trying to encode place and head direction separately. That led us to create conjunctive place and head direction cells or pose cells. And of course, the conjunctive grid cells fulfilled that niche quite nicely.
5 minutes.
Okay, 5 minutes, great. And then there's a number of others. You can add the roll dimension by adding pitch and yaw. you can just encode X, Y, sorry, you can just encode it in a straight three-dimensional cube model. And then you can go to more sophisticated measures where you have conjunctive cells as well. So there's a very large number of possibilities. And I guess I'm interested to learn from a neuroscience perspective, which are the more plausible ones. So you can do the same thing with grid cells. I'll skip through these just to note that some of these representations become very unwieldy. You need extremely large numbers of cells, which obviously doesn't happen in biology and isn't even practical in a robotics context. All right. And finally, we've been doing some work. This is led by one of my PhD students, Marvin, looking at how we can model these very compact networks found in animals like Drosophila. And we've found that some of these networks can provide extremely powerful filtering properties for navigating robots. And in a world where it's obsessed with deep learning and very complex, sophisticated neural networks, these very compact neural networks provide very good performance of great interest for roboticists. And then just finally, I guess we're doing a lot of other work. So ant-inspired navigation with this cute little Miro robot navigating many hundreds of meters using sort of ant-inspired techniques, other grid cell-inspired mapping work, and a lot of biologically inspired perception work as well. We're hiring, always interested in collaboration. These are my shameless plugs as we go through. And these are the key questions I'll sort of leave us with. Just to touch on a few, what are all of the known structures for encoding motion in above sort of 3 degrees of freedom? We know a lot of them, but we're unaware of many of them, we're sure. And one that we're particularly interested in is how does the environment, the platform, so to speak, so the sensing modalities, the compute capability, and the behavioral repertoire of an organism, whether it's an animal or an insect, affect the structure of the neural encoding of space, because it does vary somewhat. And these are some of the questions we'd love to get into throughout the rest of this conference. So with that, I think I've kept the time reasonably well. Thank you very much to the organizers, and I look forward to the rest of the conference.
Okay. Thank you, Michael. Let's see. Excuse me. A very nice talk. We have some votes going up here. So the first question is, what's the most challenging part for a neural robot to navigation or for navigation?
What's the most challenging aspect of trying to create neurally inspired robotics? I guess all of conventional robotics around navigation systems is predicated on, I guess, probabilistic formulations of navigation. So sensing is uncertain, navigation is uncertain. So mapping algorithms that inherently or explicitly represent this in a probabilistic way have been proven to be superior over the last 20 or 30 years. The problem is that the encoding of probability and uncertainty in a neural network type implementation is very challenging. Just to give you one example, in a robotic system, the traditional way to represent uncertainty in where you are is to have some sort of particle filter or representation of your location in space that gets more and more spread out over time as you become more and more uncertain. Now, that's almost impossible to do in a sort of neural network encoding of location. But one of the alternatives that we've used a lot is, rather than having a gradually increasing uncertainty of where you are, you have multiple hypotheses of where you are. So you have multiple packets of neural activity, each packet representing an alternative hypothesis about where you could be. So the ways in which you represent probability and uncertainty do vary, but there are ways to sort of achieve the same end outcome.
Okay. We have one more question here that's received a lot of votes. And that is, it's this one sort of, well, it's actually, we have two more questions here. So first is, how critical are grid cells for the robot to work well?
Okay.
Wait just a second. Wait, hold it.
Oh, sorry, keep going.
Well, it just disappeared on my screen here. How critical are grid cells for the robot to work well? What advantages do grid cells provide compared to previous models that don't use grid cells? And we have one more question after this too.
Great question. So, and once again, to emphasize, we're just one of many groups modeling these systems. I guess we're more on the robotic practical side of things. The two key aspects that we found are critical to functional performance are one, having some of your spatial cells be conjunctive. So in the case of, I guess, the deeper layers of grid cells, having some of the cells have conjunctive place and orientation properties is critical. Those conjunctive properties are what enable you to encode and propagate multiple hypotheses about where you could be located in the world in parallel. And it's very hard to do that with non-conjunctive representations. On a practical side, I mentioned this briefly in the talk, the reuse of grid cells to potentially encode thousands of places is also critical. You run out of, even with modern compute, if you're trying to map an entire country or the surface of the world for a long intercontinental journey, you run out of encoding power real quick unless you have some sort of effective reuse mechanism. And also the multi-scale properties of the grid cells have very attractive properties in terms of path planning and long-term navigation. Dealing with the short timescales of neural dynamics in a more biologically faithful model is very challenging, but we think there's a lot of potential there as well.
Okay, and we have one final question. This will have to be a sort of a short answer. This one's sort of near and dear to my heart. How do you model 3D? How does your 3D model relate to the empirical findings of 3D representations in bats, for example, from the Ulanowski lab, rodents, for example, in the Jeffrey lab, and from humans in the McGuire lab? Before you answer that, I'll just add a little infomercial from my own work. We don't really find 3D head direction cells in rats, at least in the areas where there are classic head direction cells. The cell fires just as much in the horizontal plane as it does in any of the other pitch and roll planes. We have a nice review of that in the Journal of Neurophysiology. So with that, go ahead, Mike.
Great. Just in brief, I guess, it should be very clear that we're not claiming in many of these cases that these particular spatial structures that we're trialing do exist in the brain. I guess we're having a sort of free-ranging exploration in terms of our journey into 3 degrees or 6 degrees of freedom movement. We're relatively early on. And I guess at some stage, well, conferences like this, we're going to increase the integration with neuroscientists. But it's sort of this balance between what works from a functional perspective and then what may relate back to the actual encodings that we observe in nature.
Okay. Thank you very much, Mike.
Thank you.
Yep. Okay. So our next speaker will be Stanley Heinze from the university, or from Lund University in Sweden. And the title of his talk will be The Circuitry of the Insect Central Complex Towards Understanding the Neural Basis of Oriented Behavior Across Insects. Stan, are you there? Stan, oh, there you are.
All right, and so can you see my screen?
Yep, I can see your screen.
Wonderful. Yep. Very good.
You might want to, put it on the, yep, like that. Great. Okay, so go ahead.
All right, wonderful. So thank you, first of all, for the opportunity to present my work in front of the probably largest audience I've ever spoke to, which is quite intimidating, but also really cool. And it's also the first talk I've ever given without wearing shoes, which is amazing. And so I would like to talk about the circuitry in the insect central complex. So I'll be also one of the most exotic speakers here in this crowd, I suppose, because I will not be talking about grid cells or anything like that, but rather I'll be talking about bees. And when I talk about navigation, just to get everybody on the same page, like I was thinking in terms of something like you can see here. So a bee leaves its hive, flies through a convoluted journey into the meadows and finds some food and then returns back home into its hive. And so along this journey, like essentially independent of the strategy it's using, it has to always do a certain kind of elementary decision. It always has to compare its current heading to its desired heading. And if these two angles don't match, it just has to initiate steering behavior. So it has to turn to make the current heading align with its desired heading. And how does it do that? So essentially, the model I'm working on is shown here on the right side. So what you can see is that the brain essentially creates a internal kind of representation of the current heading based on sensory information. And then it compares that to this internal kind of representation of its desired heading. And so that is based on like many more things. So previous experience, its a motivational state, its behavioral state, and like importantly, the species identity. So who you are to basically also will determine where you would want to go. And if these two directions don't actually match, you can initiate steering behavior. And so we do that with like millions of neurons, but in the insect brain, it's a little bit simpler. So that's why I stick to the insect here. And so essentially, like in my group, we're trying to answer 3 different questions. So first of all, how does the animal know where it is going? Secondly, how does the animal know where it wants to go, which is a little bit more tricky? And the third one, like essentially, how are these two angles compared to initiate steering? And so one region in the insect brain that is involved in all these 3 different processes is called the central complex. And some of you may have heard of it, some may have probably not heard of it. So essentially, it's located right in the middle of the insect brains. What you can see here is a 3D version of an insect brain, and the green bits in the middle are what we call the central complex. And that contains various subunits, which is not really important how they're called at the moment, but what is important is that it's actually an amazingly beautiful structure. And it actually consists of very regular layers and columns. So from the left side of the structure to the right side, there's always 16 to 18 vertical kind of columns. And so these are repeating elements, which will become important later on. And this region also is incredibly highly conserved. So wherever you look in the insect world, essentially, you'll find that region. And so you can see here some reconstructions of insect brains, and they all look very different. I mean, they're all kind of a little bit similar as well, but quite diverse. But what you can always see in the middle is this green bit, and that's the central complex. So that is the region that varies least amongst all brain regions in the insect brain, like even if you compare it quantitatively, which I don't have time to go into, unfortunately. But then the question is, what does this region do? So when we now do physiology, so we essentially place an insect in a virtual reality arena, and we face it with like an artificial sky, which is seen in purple here, and a 360-degree kind of LED version of the world. And we stick an electrode inside individual cells. We do intracellular recordings, and then what we do here is we rotate a linear polarizer on top of the animal. So essentially, that is kind of mimicking a situation in which the animal would be rotating like under the polarization pattern of the blue sky. And so when we do that, we get neurons in the brain that respond like this. So they have the sinusoidal kind of modulation of the firing rate. So whenever you do this 360-degree rotation of this filter, you get these cells that are tuned to specific angles of that stimulus. And this is how some of these neurons look. So they're all over the central complex, and they can be found in many different species. So I've been starting to work in locusts and in butterflies and in bees, and they're all over the place. And also in the fly now they've been found as well. And so they are located in these various subregions of the central complex. And what is important here, you see that some of these neurons actually have these input regions in very small subunits, like within this structure. And depending on where they are tuned to specific angles of this sensory information. So when you then compare cells from the left side with cells from the right side, you can see that the further you move along the structure, the further this angle of heading representation changes. So you essentially have an ordered array of head direction cells, or heading direction cells, if you want. And so another cue that is like interesting in that context is actually velocity of the animal. So what we do is we have the animal in this virtual world, and then we can basically mimic the optic flow environment as if the animal would be flying forward or flying backward as a control stimulus. And when you do that, and we do that always in kind of bouts of 3 seconds here. So the first green line is when the stimulus is switched on, but not moving, and you can see that there basically is like nothing happening. And as soon as the stimulus starts to move, you can see that when you simulate front to back optic flows of forward flight, you can see that that cell is highly activated, and like nothing happens when the opposite is done. And so when you now do that with various speeds, so you can see here that is a plot of the forward velocity against the mean activity of that cell, and you can see that the faster you move, the higher the activity of that cell becomes. So that's essentially a speed encoding neuron. And so that's how they look, and they, surprise, also go into the central complex. So they would encode speed of the animal based on optic flow information. And so that is really interesting, because that's exactly what you need to do something called path integration, right? So you fly out on a convoluted journey and you want to come back in a straight line. So you basically update your position estimate each point like of your convoluted journey, and as soon as you decide you want to go home, you can do that in a straight line because you have this accumulated information. And all you really need to do is actually measure all your directional changes. So you need a compass, and you have to measure how far you went onto each of your flight segments, and so you need an odometer. And now bees in particular, they use visual cues for both of those things. So as you've just seen, there's actually polarized light that is used to basically measure like which direction you're facing, like within the global frame of a reference of the sky, and they need this optic flow information to measure speed. And if you integrate speed over time, you end up with distance. And so that is very convenient because we have both of these things now in the central complex. And so that was very intriguing because we thought, okay, so maybe there is a path integrating circuit within that brain region. So now there's a little problem, though, that even though it's in the same brain area, it's not exactly in the same place. So we have these skylight compass information actually in these highlighted regions here, and we have this optic flow information in another region, like within the same structure, but not exactly in the same place. So how are these two things integrated? So that's what we'd have to find out. And so one strategy we wanted to follow up is to see who's postsynaptic, or basically downstream of the speed neurons. So we did something called serial section block phase electron microscopy, and we basically reconstructed all the neurons found in this little region here. And then when you zoom in a little bit, you can identify who is actually really postsynaptic of these speed neurons. And so the gray blobs here, they're these kind of axonal terminals of the speed neurons. And so what you can see here in red is actually the active zones. And when you then see who's on the other side of that, you can find these small kind of colored segments here. And so if you then trace them back to the point of origin, you can see that these are so-called PFN cells, or CPU4 cells, but the name doesn't really matter. But it's important that they actually are cells of the central complex, and they are actually these columnar cells. And this is how they look. So you see these in a color here now, and there's a whole set of these cells, and so all of them receive input from these speed neurons. So all of them get this optic flow information within this region here, which is called the noduli. But at the same time, they also get input from this region up here. And if you remember from earlier in the talk, that is where all these kind of directional cues are actually represented. So that's this map of head directions. And so each of these cells now receives input from a specific head direction. And so if you now assume that this information, which probably can actually gate the optic flow information, and then if you additionally assume that there's some kind of integrator within this neuron, you'd have basically a set of direction locked odometers. So you can imagine that whenever you fly in one direction, you accumulate optic flow information, and you're actually raising your kind of background activity within that cell. As soon as you turn, you're accumulating in another direction. And so over the turn of your journey, you essentially establish a kind of integrated version of your outbound journey, which you then can just invert and fly back home in a straight line. So basically, the entire journey would be represented as a population code within this set of CPU4 neurons. Now, that's a nice claim, and we did try to model that, actually together with Barbara Webb in Edinburgh and her student Tom Stone. And so essentially what we did, we drew out all the connections we could find in the insect central complex, which is quite messy. And then we put that into this force-directed graph diagram, and what actually popped out surprised even us, because it was exceedingly beautiful and very simple indeed. So you could find this same cell the mass on the left side is essentially like identical to this nice ordered array of 3 concentric rings. And so what you have here in the middle is actually a ring attractor circuit, which is located in the proto-cerebral bridge here, and it basically serves as a kind of a representation of the current heading. In the second layer, you'd have a desired heading, which is essentially the memory ring. And in the third ring, you'd have a circuit that actually samples the activity within this inner ring and compares it to the activity in the middle ring, and then initiates steering behavior if these 2 bumps of activity are not aligned, in theory. So if you actually model that, you can actually put this circuit in a virtual agent, and you can have it fly through a randomized journey, and you can observe how the activity within this inner ring is always aligned with where the bee is facing. And then the bee moves away from its home, and you can see that activity has accumulated here on the right side, actually where home is. And then if you actually instruct the circuit to now steer the bee, and that has happened here at the point, and now the green line is actually this circuit steering the animal back to where the circuit thinks home is. And as you can see, it steers quite well, but there's no stopping signal. And then as soon as it's overshooting, it starts to accumulate a new home signal. And so that starts to essentially loop around home, which is exactly what insects do. And that's quite cool, because we didn't program that into the model. It just is an emergent property from that model. And so that was quite encouraging. And we also implemented it on a robot, and it also finds back home. So when you now think about what this circuit tells us, like one of the most important take-home messages for me is that the details of the anatomical projection patterns of these specific sets of neurons within the central complex, they are what defines the computation, right? And so that is really, really interesting, because we then can draw this direct connection between the anatomy and the physiology. So when you wrap this up a little bit, is that we now have a circuit which has a separate representation of the current heading. We have a representation of this goal direction, and we have certain cells that compare the 2 to initiate steering. So essentially, we have a circuit for elementary navigational decisions. And so how they are chained together will then form the strategy of navigation. But because that elementary decision is required for each navigational behavior, it should be present in all insects. So that's what we do next. So we're actually comparing across species. And so that's what we actually currently work on. And essentially, we ask the question, how conserved is the circuitry across insects? And so we're starting actually with the bumblebee central complex here. And now that we have the entire ground truth, so the entire connectome of the fruit fly, we can actually see which parts are actually there and which parts are the same and which parts are special. And so this is just a very short version of this, because it could go on for forever about that. But essentially what we find is that there are certain, all the core conserved, or basically all the core computational units that you find in the fly brain, we also find in the bee, and that they're completely identical. But then additionally, we also find other cells which are a little bit more special. And so we have those just a few illustrated down here. And so there are some cells that are found in bees which we have not yet found in the fly. And so the question is, maybe this additional complexity of bees' behavior is maybe due to those additional cells, which we don't know yet, but it's a good start to look into things. And so basically, to wrap that up, the core cell types are the same, but more cells may exist in more complex species. And so one of the important things that we learned from the model is that the general projection patterns, they are actually the core for doing the computation. So they define the computations. And so let's look at those. So basically what we see here is that there's certain rules as to how these cells map like onto each other, essentially. Let's not go into details, but let's compare to the fly. And so even though there's like about 300 million years of evolution in between those, you can see that these patterns are completely identical. They're absolutely the same. And that is really astonishing, I think, because it tells you that these core computations are exactly the same, and they're highly conserved across all these ages of evolutionary time. And so now we have the conserved core computations, but still we have like super highly diverse behavior. So how does the same circuit generate all these like various behaviors? And so then we have to compare more species. And so what we then did is we developed, or actually my student who is here, actually Marcel, he developed a new way to actually get into the actual kind of connectome of this brain area. And what we did is we have a multi-resolution approach here, because the structure is quite large, even though it's within an insect. However, the central complex itself, like in a bee, is about the same size as the entire flyer brain. Being only one research group, we can't really do the whole thing at synaptic resolution. But what he does now is he developed this approach in which he gets one like overview stack, which is about at like 20 to 50 nanometers of resolution, and that we can use to generate a projectome. So actually the entire kind of the entire set of neurons and how they actually map between these different subunits of the brain region that we're looking at. And then we have individual parts in here that we can do at synaptic resolution. And because of the modularity of this brain region, we can then extrapolate like into the entire connectome. So we have this kind of overall idea plus actually local synaptic connectivity. And we do that in 6 species at the moment, and a few of them are like already done, and we are working on the next one. So that should be finished by the end of the year, and then like lots of work will be done with actually all the tracings. So that's just a glimpse of like what we will do. And so the conclusions I can come up with is essentially the central complex and its accessory neuropils are the most conserved neuropils in the insect brain. The central complex encodes the current heading of an animal, or of the insect in that mode, and it is also involved in encoding the animal's speed. And basically, these two things are actually wired up in a way that would allow this brain region to serve like elementary navigational decisions. And that is probably a foundation that is really highly relevant for all like oriented behaviors. And so the involved cell types and the projection patterns are highly conserved to 300 million years of evolutionary distance between flies and bees. And so we have a new imaging strategy that now hopefully will enable us to do this in a much more like across species way, and hopefully like allow us to identify how like kind of different behaviors emerge from this extremely conserved circuitry. And with that, I'd just like to thank my group and all the collaborators we have, and all my funders, of course, and then also you for listening. And that's it. And thanks.
Thank you, Stan. I actually made a little mistake there in terms of my own timer. So when I said 5, you really had 10 minutes left to go. So you did fine.
Okay.
Sorry about that. That's my fault.
I forgot to switch on my own clock, actually. And I actually didn't even hear you saying 5 minutes. I have to admit.
Oh, good. All right. So we got a couple of minutes. We're right on time. We got a couple of minutes of questions here. And as I'm seeing the tallies come in, the first one is path integration and head direction attractor dynamics in rodents is thought to depend critically on the vestibular cues. Is that true for these signals in insects, or just optic flow is enough? These bees are head fixed.
Yes, so basically it's all head fixed and we don't actually know, but what we know from flies is that they do use self-motion cues as well, and it's not all about vision. So they have like kind of other modalities as well that they use, but they're much more like error prone. So if you actually watch your kind of turning movements at all times, you accumulate kind of rotational error. And so actually using these more global cues is much more reliable. And so because actually, so if you go out further, you get least error. And in terms of distance estimate, what you'd have is probably optic flow only if you're flying. But if you are walking species, actually like an ant, you're actually kind of integrating the steps you're making. And so there's some various strategies. And the neurons, though, are highly conserved. So probably they look into the same circuit in there. So they feed into the same circuit. And so that's why we're actually comparing kind of ants and bees as well. That's basically one of the big questions we have, as to if that's the same or not.
Okay. Next question is, any path integration system will inevitably accumulate noise. How does the circuit implement error correction?
Well, it doesn't, but it is a couple to this emergent search behavior in the end. So so it's obviously kind of limited in how far you can actually go out before you can't really go back. But the behavior is remarkably accurate. However, it's always a trade-off between how precise you are and how far you can actually go out. And so the system can work with surprisingly noisy inputs. So that's actually why we really put it on a robot and see if how it works with real-world input. And we did that in a classroom thing with like the walls being closed on one side and like this kind of people walking by, and it still did extremely well. And we were quite surprised by that, but somehow the errors just more or less like average out, like at least in the robot that we tried. And so that was a phone on wheels, which is not quite like an insect. But then actually Barbara has now actually put the model on a drone, and it works equally well. So that's quite remarkable, and we don't really know why. And we're actually starting to basically develop it into like a nanotechnology-based like decision-making circuit that we also want to actually really put into nano drones as well, and see if we can actually take over the world with those kind of super robust behaviors.
Okay, and maybe one final question. Try to keep this answer short so we keep on time here. It was a fantastic talk. That was the comment. Can the memory network store more than one goal location?
Not with the model we have, but with insects, there's probably the possibility that they can, especially with bees. And so there has to be some way of outsourcing the information. And the mushroom body, which is like the big memory center in the brain, that's the obvious place where that should happen. And there is some ideas of how that can be. And I don't have time to go into that, but there is a possibility to store more than one location and just recall them actually back in and do more kind of complex ways of vector navigation and route optimization and things like that through that mechanism as well.
Okay, great. Thank you very much.
But thank you for listening.
See you later.
Right, see you.
Bye. Okay, our next talk is by Mark Brandon at McGill University in Canada. And the title, hi Mark. And the title of his talk is Population Dynamics of the Thalamic Head Direction System During Drift and Reorientation. Mark. You want to just test your microphone, make sure your microphone's on?
Let's see.
You're good. We hear you.
I'm good? Okay.
Yep.
And let me...
I'll try to get the timing correct on this next one.
Okay. Thanks, Jeff. And can you see my laser pointer?
Yes.
Okay, great. So first of all, I want to say hi to everyone watching, and thanks to the organizers here. I'm obviously quite sad that we couldn't do this in person and see everyone in Italy. But I'm also happy to keep everybody safe, and I think this is a great opportunity for trainees and people interested in navigation and the neuroscience of navigation across the world to participate here. So I know it hasn't been it can't be easy to organize this on such a big scale. So I think I speak on everyone's behalf when I thank the organizers, Jeff, Thomas, Mayank, Josh, Lori, and of course, Suzanne. So thank you for inviting me, and I'm happy to talk about and share my research that my lab has been up to. So my title of my talk is Population Dynamics of the Thalamic Head Direction System During Drift and Reorientation. And so I will be introducing mammals here. So here is just a brief overview that Jeff had shown in a recent review paper of the brain's navigation circuit. So in green here is the sort of the vestibular inputs up into the thalamus. So this is where you see angular velocity neurons. In blue in the thalamus, and then up into the cortex is where you start seeing head direction cells. And of course, this feeds into the famous entorhinal cortex grid cell system and the hippocampal place cell system. So what I'll be talking about today is all in the thalamus, so the anterior dorsal nucleus of the thalamus. And so you see vestibular information is coming from the medial vestibular nucleus and then up towards the head direction system. We have visual information coming top down from visual cortex into postsubiculum and retrosplenial cortex, feeding into the anterior nucleus of the thalamus. And what my lab is interested in, how do these multiple information streams converge in the thalamus to create a stable head direction system, both based on vestibular inputs, movements of the animal's head, as well as information coming from the environment, mostly visual information and visual landmarks. So everybody here probably knows what a head direction cell is. These are neurons tuned to where the head is facing. So here's 4 examples of head direction cells. And this is typically modeled as a ring attractor network, where neurons that have similar directional tuning are tightly connected to each other. They inhibit cells that are further away on the ring. And this is sort of a one-dimensional manifold in which the head direction system has been modeled. And of course, angular velocity inputs are modeled into this with angular velocity cells starting in the DTN up into the LMN before the anterior nucleus of the thalamus. And these are neurons that are updating the system as the animal turns its head in a clockwise or anti-clockwise motion to sort of move the activity along the ring attractor. So I just wanted to introduce these topics before I continue on. So visual information is also coming, from the cortex, from visual cortex on down, and it's shown that head direction cells, in fact, do need this visual information as head direction cells drift in darkness. So here's a paper from Valerio and Taube in 2012, where they're recording from head direction cells in the animal's refuge in a path integration task, and this is where you can see the tuning of these head direction cells shown in black. And then when the animal goes into this open field environment where he needs to perform a path integration task in complete darkness, the head direction system drifts. So in this case, you can see these same head direction cells drifting, and that's what's shown in these red curves here. So when there's no visual information, the system starts to drift. When the animal gets back to the refuge, and this is reported in this Valerio and Taube paper, and this is what's motivated the experiments I'm going to show you today. When he gets back, there's actually a reset where the system will sort of reset back to this black curve. The animal has sort of re-engaged or reoriented to the environment, and now the head direction system is reset. And then finally, one of the first findings also in Jeff's lab is that head direction cells are anchored to visual cues. So when they're recording an animal in a cylinder and you rotate a visual cue by 90 degrees, the head direction tuning curve shifts by 90 degrees. And you see this here in this one example of a head direction cell. So visual information does reset the head direction network. It's important for stability in the network, and without it, the system starts to drift. So just sort of a schematic of this, you can imagine an animal in an open field environment, and this star is indicating a visual cue in the environment. And these are what the head direction cells would look like. So you could have 4 head direction cells coding different angles of this environment. You could do some basic decoding of this, where the measured head direction angle would correspond well with the decoded head direction angle. You could decode where the animal is facing just based on a small population of head direction cells recorded. And then say the next day you rotate this cue 90 degrees. Here you could have the measured head direction cell, the measured head direction activity is actually now going to be off by 90 degrees if you're using a decoder based on this first day. So in a way, the system is reset by this shift of the Q. And so you could imagine a decoding analysis would actually show this reset of 90 degrees. And I'm illustrating this only because this is the type of analysis that we'll be using later on in the talk. So our goals here in my lab for head direction cells is to try to understand the dynamical structure of the attractor network and how this model of head direction cells could explain how drift and Q reset would fit into the model. We sort of have this guiding hypothesis that to allow for resets and drifts to happen, the network should account for these by changes in energy levels of the head direction system. And thus, this should become a multi-dimensional structure, not physically, but maybe in a manifold space, where the system would consist of multiple energy levels that would allow the system to rotate faster through head direction space. And you could imagine these trajectories through this disk space or this two-dimensional manifold space. So to get at this, a student in my lab, Zaki Ajabi, a very talented graduate student in my lab, just finishing up now, used the UCLA Miniscope. This was developed by Daniel Haroni, now still at UCLA, where we injected a virus to express GCAMP in the ADN of the thalamus here. This is where the head direction cells live, and we put a long relay lens down into the thalamus to record from as many cells as we could, so that we could get a population understanding of what neurons are doing during drift and during these resetting events. So of course, here's just some tuning curves of head direction cells that we can record with the Miniscope, and this technique just allows us to record enough cells to cover all directions. So in this case, in this recording, there's 111 head direction cells that are spanning all possible directions that the animal would face. And so we can do fun things like paint what the anterior dorsal nucleus of the thalamus looks like based on the neurons are colored by the direction that they're coding for, and you can see there's really no topography, and this is true across animals. So that was our first little fun with the Miniscope to see if there was topography, and what the tetrode experiments did suggest turns out to be true. There seems to be no topography in the ADN. So because we're able to record from large populations of head direction cells, we can do some very nice decoding of neural activity. So here in this bottom plot here, the measured actual head direction of the animal is in blue, and the decoded head direction is in red, and these are overlapping quite nicely. This is a novel Bayesian method developed by one of our colleagues, Shuai Wei. And with this, we can get sort of a median error of about 10 degrees at which the animal is facing. So it's a very nice decoder covering all angles that the animal could face. And then a second analysis that we were doing was this sort of dimensionality reduction technique that allowed us to project neural activity into a two-dimensional ring. We can talk about the details of this later, but the idea was that if we can project it into a ring, then perhaps the radius of this ring would tell us something about these lower energy states that the system might go into to create shortcuts through this ring attractor. So a couple of the questions I wanted to cover in this talk. The first is, does activity rotate around the ring attractor during a visual resetting event? So I'll just get right into that. So the way that we did this was to put an animal in this chamber where he's just sitting on the small pedestal. He's not really doing anything. He's free to move around. And then he's surrounded by a 360 degrees LED screen that can project these white cue cards onto him. So the rest of the screen is black, and then we can project a white cue card. And so the experiment was sort of go like this, where we had this initial cue followed by 2 minutes of complete darkness. And then when we turned the cue on, instead of just turning it on where it initially was, we'd play this little trick on him where we'd change it and rotate the cue by 90 degrees. And then we just continued to do this to really induce these resetting events. So during darkness, maybe we'd see some drift, but then when the cue came on, the system would need to snap back to align to the visual cue. So we thought that maybe there's two possibilities, and these were really outlined by this great paper by Michael Zagaro back in 2003, where there's two possibilities of what the attractor network could do. It could either slowly rotate, or even maybe quickly rotate, when the system is reset by a visual cue, or there could be this abrupt transition where the state that's currently on would start turning off, and then the state that would be driven by the visual cue would gradually turn on. So there'd be this competition on different sort of places on the attractor network itself. So is there a rotation, or is this abrupt transition? And so here's what the data looks like. This is the difference in the decoded head direction versus the measured head direction. And we're decoding based on this this initial cue segment, so this beginning part of the recording. So you can see in the beginning, when the cue is there, you see very little drift. That's what's plotted on the y-axis here is drift. So there's very little drift. It's very stable. He enters darkness. We see maybe a little bit of drift. And then after 2 minutes in darkness, we rotate the cue by 90 degrees. And you can see this abrupt shift in the network to 90 degrees. And then again, we do the 2 minutes of darkness, and then again, an abrupt 90-degree shift. And we continue doing this until we get back to the initial cue. So here's just a couple more examples of this. We can go in either direction, but this happens every time. Every time we rotate the cue by 90 degrees, we get this very nice reset event in the network, where the network starts to represent as if the head direction system has needed to rotate 90 degrees. And what you see is in the data, even just some examples here, we see some events that are quite fast. Here's a reset event. As soon as the cue came on, it just took 5 seconds for the system to reset. Here's one that's still fast, but maybe it took a little bit longer. This one's 12 seconds. And here's a much slower resetting event. This one took 29 seconds. And so we saw both fast and slow resets. Here's just a nice depiction of what this looks like. So here's a sort of zoomed in, this very fast reset here, where almost as soon as the cue turns on, we see this the activity rotate. And then here's a very slow reset, where it actually takes quite a bit of while when the cue turns on for it to start moving. And when it's moving, you can see it's actually moving relatively slow. The x-axis here is true. This is 90 seconds. So it's taking almost 80 seconds for the system to actually rotate 120 degrees in this case. So what this means to us is that, yes, we do have these fast resetting events that look like maybe they're abrupt, but more often we see these slower resets, where as soon as the cue comes on, even if the system needs to rotate 90 degrees in order to lock onto that cue, it takes quite a bit of time, and the system actually rotates itself in order to eventually lock onto that cue. So there is a rotation. Activity does propagate around the ring attractor in order to reset to visual cues. So then we wanted to ask a little bit more about the speed of reset, because we saw some fast, some slow resets. And we wanted to ask if network gain, or maybe this two-dimensional manifold, or the one-dimensional ring attractor might be a two-dimensional manifold, where network gain could predict the speed of the reset. And what I mean by this is when we look at the two-dimensional or the one-dimensional manifold here during baseline, it's very nice. So this is just this initial few minutes of recording. But then when we look at the whole session, you see the system starts to collapse close to the center of the circle. So there's definitely this dimension along the radius. And perhaps this is the network taking shortcuts through this two-dimensional manifold. And so we looked at this carefully, and we saw with fast resets, we actually saw that the activity would collapse towards the center of the manifold, as if it's taking a shortcut. Whereas during slow resets, activity was more bound towards the ring of the network. And so we did some modeling, and we realized that this could result from incoherence. Either the head direction system is no longer coherent. Cells that are coding north and south are both being active and sending the system to the center. That didn't seem to be the case. It seemed to be really gain modulation. So just overall network activity levels were causing these shifts in the radius. And so what we did is we sort of modeled gain as just a relationship between how much, how much do we need to multiply the tuning curves of a neuron in order to match the current firing rates. And so we see that this gain factor, which is how much we need to multiply the tuning curves, actually corresponds well with the length of the radius, with the radius of 1 just being the edge of the ring attractor. So we started looking more carefully whether if gain amplitude was predictive of reset speed. And so we broke the data into 2 groups. One would be slow resets, or this would be a fast reset or a slow reset. So this is the amount of drift over time. You can see. Sorry?
5 minutes.
Okay. So we have these slow resets that are in blue. So here it's taking over 20 seconds to reset 90 degrees, and in purple this is less than 20 seconds. So this we consider a fast reset. And when we looked at the gain of activity in terms of basically the overall network level, we saw that these slow resets in blue decreased their activity to an extent over a long period of time, whereas the fast resets had to decrease their activity quite substantially. So there's a very big difference here where gain amplitude, the more the gain is reduced, so the more reduction, the faster the network is able to rotate. And so here we see a nice correlation between the average reset speed and average gain amplitude. And so we could model this just by, this is just a very simple model of the head direction network, and just by adding this additional factor where we could modulate the gain of the head direction ring attractor directly, we could model the system and then very nicely replicate the types of drifts we saw. So in blue here is the true drift, and in red is the simulated drift. And with a logistic regression algorithm, we could classify these quite accurately. So I want to skip number 3 because of this timing warning I just got. But then the last thing we noticed in the data that was quite interesting is that in this 4th dark period, as we're rotating the cube progressively, we noticed that the drift direction was biased in the same direction of the resets. So it almost looks like it's predicting where the next queue is going to come online. And so when we looked at this more carefully, we saw that there was quite a bit of drift in a particular direction during this last darkness period, as if it's been biased by this constant rotation throughout the experiment. And so we said, is it biased by the direction of the previous resets, or is it an attraction back to the initial queue location? And so we did one last experiment to test this, just to see if visual stimuli prior to darkness would bias the drift direction. So we had five minutes here of a stable queue location, then some darkness, and then we rotated the queue very slowly, so 1.5 or 3 degrees per second. This is not enough to get dizzy, for 5 or 7 minutes. And then we turn the lights off and look to see if what would happen after we've rotated the queue. So this is what the data looks like. Here's the stability. There's very little drift. In dark, there's a little bit of drift. And this is what the drift looks like when you're rotating the queue, so that it's very nicely, the system is very nicely locked in following the queue. And then when you turn the lights off, surprisingly, the system continues to drift in the same direction and the same speed at which we are rotating the queue. So this here is in complete darkness, but the system continues to drift regardless of what the animal is doing. Sometimes the animal is moving around, sometimes he's sitting still. And very often we notice that the system would stop drifting as soon as it got to its internal queue location. So when the queue was in the initial spot that it initially was. So here's many examples of that. You can see sometimes there's very persistent drift, sometimes there's the stabilization around where the initial queue was, and sometimes things get rather complex and rather hard to understand. But at least we do know that this the drift is biased by the prior visual cues. And here we've even quantified this. So the cue velocity prior to when the lights are turned off matches the drift angular velocity when the lights are turned off. So just to sum this up quickly, I just showed number one, that yes, activity does rotate around a ring attractor during visual resets. So this does suggest that visual information does have access to the angular velocity elements of the head direction system. And the network gain does predict the speed of the resets. And so to allow for resets and drifts to happen, the attractor network accounts for changes in energy levels or gain modulation of the HD system. And thus, this is more like a disk-like structure in manifold space. And I skipped number 3, which was quite interesting, but just in the interest of time, I went straight on to number 4. And is drift direction biased by prior visual stimuli? Yes, definitely. This suggests a type of short-term plasticity between visual information and the angular velocity component of the head direction system. So just my acknowledgements quickly. I want to once again thank Zaki Ajabi, the graduate student, PhD student in my lab, who ran all these experiments and has been instrumental in getting these analyses to work. He's worked very closely with Liam Paninsky's group, and specifically Shueshen Wei, who's now at the University of Austin, who had worked with Liam Paninsky. So thank you very much for his help, and as well as to Alexandra Kainoff, who has been also very instrumental in getting this project going. So thank you for your attention. I hope I didn't run over time, and I'm looking forward to your questions.
Thank you. So I'll just say it's nice to see a confirmation that you didn't see much topography, at least in the anterior thalamus, which sort of confirms a lot of previous studies, but that's nice. Okay, let's see here. The top vote getting question. I feel like I'm a quiz show person here reading off the answers. Okay, does the reset duration depend on past rotations?
Does the reset duration depend on past rotations?
Yep.
So I'm not sure what you mean by the past, either within experiment or over days. I can say that it doesn't seem to. So even within the same animal, within the same experiment, the research durations seem to be randomly distributed, and across time, they seem to be randomly distributed. The thing that we do notice across time that seems to change is that after the first initial cue, that first darkness period becomes more and more stable. And after we train the animals for months, almost all darkness periods become very stable. So it seems like they're latching on to another reference frame of the environment.
Okay, I think the question was getting at was like between the fast and the slow resets. Was there a difference on, let's say, the first week of doing the experiment versus 8 weeks later? Was there a higher frequency for one?
No, it doesn't seem to be. No.
Okay. All right, we have time for one final question here. We have a tie. Is there any relationship between reset speed and the gaze direction of the animal at the time of the cue onset / offset?
Yeah, that's a great question that we've been analyzing in detail. It does seem so. It seems as if the animal is facing the cue, the reset is faster, but it's not always the case. And even when he's facing 180 degrees away from the cue, it still can be fast. So I do think that there must be some kind of attentional modulation here, and that's what this data suggests.
Okay, and there's actually one more that's been getting some more votes, so maybe you can answer this one quickly. When there is no topography of preferred head direction, how is the right connectivity for the ring attractor determined?
Yeah, so this is an age-old question. And certainly the answer is that the topography would have to occur in synaptic space. So regardless of where the cell bodies are positioned, that head direction cells with similar tuning would be connected to each other synaptically, even if they're not right next to each other.
All right, thank you very much, Mark.
Great. Thank you.
See you later. Alessandro, there you are. How's it in Italy?
Very, very nice. How do I share my screen?
Okay. So our next speaker is Alessandro Treves from the International School of Advanced Studies in Trieste, Italy. And the title of his talk I thought was a great one, although I have no idea what it's going to be about, is Outside the Lab, Disorder and Frustration. Hopefully there's some navigation in it.
How do I share my screen?
Hello, Alessandro, can you hear me?
Yes.
Perfect. So hover your mouse over your face and you should see a little menu pop
Okay,
up,
yes.
and then you can click share screen.
There you go. Looks good. I'll let you know there's 5 minutes left.
Okay, so you can see the screen. Good.
Yep.
Nice to, thank you for the organizer. Nice to welcome you in Italy, although you're not going to see the landscape of Cortina, but just my face. What I want to discuss today are the implications of the discovery of the grid cells, which have been taken to imply that a specific form of mental order can emerge spontaneously. And in particular, the fact that when we put a rat in a new environment, it appears that the grid cells reproduce the same map, suggests that there is a kind of universal metric. But an implication of that would be that this grid cell system has no memory. If it cannot distinguish one environment from another because it acts in exactly the same way, then it has no memory for which environment the rat is in. And this was quantified in a study by Marianne Finn et al., where the information about which environment the rat is in was extracted from the coactivity relations between grid cells and place cells. And the essential result is that only in place cells you find these red dots. You find that you have the information which enable you to distinguish which environment the rat is in, not in the grid cells. So that, in a sense, resolved the competition between the two narratives about what the hippocampus does, whether it's memory or space, in that it shows that only in the hippocampus there can be spatial memory. So the spatial computations are kind of outsourced maybe to the entorhinal cortex and other surrounding structures, and maybe grid cells have only a supporting role in navigation. But does it have to be so? Is it really this finding of apparent universality? Is it something that the system cannot do otherwise? Or is it possible that a network of grid cells could in principle store distinct maps of different environments? So this is something that we looked at with friends in Paris using not one but two very different mathematical models. And I will not tell you about the analysis of the models, but just the result of the two models that are different, but they essentially agree in allowing us to conclude that whereas for square grids, which are not what found in the rodent brain, one would have a very low capacity, which with the appropriate numbers for internal cortex would amount to having just essentially one map. So it would be a universal map by per force, by the inability of the system to produce more than one. Instead, with the hexagonal maps, the capacity is much higher, and the system could express of the order of 100 different maps. So in the grid system, there is potentially the ability to encode different maps for different environments. This was, we also looked at with the computer simulations that are useful in supporting the analytical calculation, but the conclusion remains that grid units could in principle express orthogonal metric representations of several environments. But then, do they do that? Well, the idea of a universal map has been having trouble from a number of different studies. One that was already mentioned is the study of what grid units are expected to do in 3D space. There is a theoretical expectation that they would produce a crystal-like structure, but this is not what is found. These are results from our models, but the forthcoming results from Nahum-Ulanovsky's lab show that the units that would be expected to be crystal-like grids in 3D, they have multiple fields, but the multiple fields show no long-range order, only local order. So it's quite a different arrangement from the one that would be expected theoretically. But also in 2D, in environments with a non-standard shape or very large environments, one can see broken or bent grids. So suggesting that this idea of the universal map that applies unchanged to all environments may be not correct. And maybe we should consider the fact that all these experiments have been, most of the experiments have been done by placing rats in this empty flat box, which are quite an artificial environment, also for humans. If we take a natural environment like Jerusalem, it was, before human intervention, it was full of ridges and valleys, which were flattened only when humans built on it. And one could ask, what would be the grid map of a natural environment with ridges, mountains, and valleys? We tried our model for the emergence of grid maps in situations which are particularly simple when the curvature is locally constant, either positive curvature like on a ball, or negative curvature like in one of these hyperbolic surfaces. And our model for grid cell formation would predict that you can have a beautifully symmetric grid, but with a coordination number different from the standard 6 of the hexagonal grid. It could be lower, like 5, in regions of constant positive curvature, or higher, like 7, in regions of constant negative curvature. If that is the situation for isolated grid units, what happens when you consider several grid units that are talking to each other? What happens to the coherence that we can observe among grid units on a flat 2D environment? Well, if we take, for example, grid units on a sphere, these are, again, are simulations with our model, we find that when we have we run a simulations, not all grid units or would-be grid units produce the same perfectly symmetrical grid, which looks like a soccer ball with 12 fields on the sphere. Sometimes there are 13. So this already tells us that there cannot be coherence between grid units with 13 or 12 fields. But even if we take two grid units that are close to ideal soccer balls with 12 fields, what we have is that they can be related or mapped into each other with a so-called Euler rotation. One can rotate a soccer ball into another arrangement, and one has another soccer ball with a different orientation. But that's a Euler rotation is not a translation. That means that the relationship between pairs of fields changes from pair to pair. Some fields are far from each other. Some, those that are close to the poles of the rotation, are close to each other. So the relation between different pairs of fields is different. That means there cannot be coherence. There cannot be phase coherence on a sphere, and that's true in general of any curved surface. Grid units cannot be coherent on curved surfaces. And that makes us ask what will happen in a natural environment. Like here is the den of a kangaroo rat, which I like because it's particularly spaghetti-like. But the point is that these natural environments have curvature, and this curvature can be measured from measurements of environments. Here is an environment of a Norway rat that has been measured in detail. So this environment contains portions, those in green, that are quasi-flat because they're close to being cylindrical, but many other portions, those that are not in green, that are curved. And one can show that one would expect an hexagonal grid structure only in a region of close to zero curvature. So here on the x-axis, there is the ratio between grid spacing and curvature radius going towards the positive direction for positive curvature and the negative direction for negative curvature. On the right, one expects if there are symmetrical grids, they would have lower coordination number than 6, and on the left, they would have higher coordination number than 6. And one would expect hexagonal grids only in a narrow strip at the center of this plot. So what happens outside this narrow strip? It's a bit a situation like if one looks at cortical-cortical connection, where there is a well-defined coherence in the direction of the fibers only between the two hemispheres. And then if one goes to the right or to the left hemisphere, it's everything goes in all directions. There is a loss of coherence. So we are looking into that by simulating a realistic environment that is a work that I will present at some other point. But just for today, to conclude, I want to tell you a few results that can be derived by looking at what happens on a sphere as a very simple model of a curved environment. So here you have one key result, that if you simulate the emergence of the fields of a would-be grid unit isolated without collaterals that allow for interactions with other units, different units can be more or less neatly mapped into each other. This is what you see in panel B. But if you turn on the collateral connections, make the units interact, you see that each unit becomes less perfectly symmetrical, less of a soccer ball, and different units cannot be mapped precisely onto each other, even with the Euler rotation. There is always some jitter in the position of their fields. So what happens is that the interactions through the recurrent collaterals, instead of producing coherence, they destroy the coherence between different units. That's an effect of curvature. and it applies in any curved surface. So coherence is impossible in curved environments. Another effect is that incoherence leads to protracted instability. So in a simulation, we see that the position of the individual fields continues to change over the course of the simulation, whereas in a 2D environment, in a flat 2D environment, it's quickly stabilized. There are more results that I skip over, just showing you something which looks a bit mysterious, but it's a plot of this clustering coefficient, which is a quantity that can be taken as an index of how a system of would-be grid units that we looked at as their fields emerge on a sphere, how is in the range between being fully coherent and showing uncorrelated grids that are not interacting and have no relation to each other. So what we find is that when we have recurrent connections that allow the units interact, the units try to form some degree of partial coherence, and that is expressed by this clustering coefficient having a scaling relation with this parameter that I have no time to explain what it is, which is exactly intermediate between that of the scaling of a fully coherent system and the scaling of units that are not interacting with each other. So this scaling, which is 1 over square root of this parameter, suggests that there is a kind of specific phase, is a new phase for this type of biological matter, the grid units, that is intermediate between the coherent phase that we can find only on a plane, on a flat 2D surface, and the uncorrelated phase that we have when the units do not talk to each other. It's a phase of partial coherence with interesting physics properties, but which has implication, we think, for the way this system can be used for navigation. So these implications we have no time to look at, but I want to conclude with this cartoon of a real rat environment, just to make the point that how a system of grid units may contribute to navigation in this real environment could be completely different from the inferences we can make from observing grid units in an artificial lab environment. So outside the lab, there is disorder. I'm not sure there is also frustration. I think rodents are quite happy in their natural environment, but I think there is, importantly, disorder in the grid system. Thank you very much.
Thank you, Alessandro. Okay, let's see some questions here. We've got a few minutes. All right, the highest vote getter. Here we go. Has anyone tried to record spatial cells in such tunnel environments, or even just on a non-flat surface?
I think is going to come soon. There are these experiments with bats in Nahum Ulanovsky's lab, where bats fly in long tunnels. And I think in principle, similar technology could be used in other realistic environments. But I guess with rats navigating tunnels, there are practical hurdles that have to be overcome. So in a realistic rat, then a tunnel is the size of a rat. So the recording equipment could bang in the walls at any time.
Okay, another question. Does the incoherency of grid cells, whoops, hold it. Does the incoherency of grid cells in non-flat naturalistic, I guess, environment imply that grid cells are not useful for large-scale navigation in real world?
I think they might still be useful, but we need to sharpen the tools to understand what is really a new phase. We should not rely in our models on the long-range order that we assumed was there from the experiments in the lab.
Okay, and lastly, and I like this one too, what's the implication for humans as well as rats living in Manhattan with its grid-like layout?
Maybe there is a special phase for New Yorkers,
right?
And people in Torino, similar layout that would not apply in Trieste or Florence.
Okay. Oops. All right. I think that's it. All right. Thank you very much, Alessandro.
Thank you.
Okay. See, we've been all over the world this morning. We've gone from Australia to Sweden to Canada and now back to Italy. So modern technology lets us do that. We're pretty much a little bit over time, but we're okay, I think. And we'll sort of take a break now and reconvene in about 20 minutes. Thomas, did you want to say anything?
Okay, I think, can you hear me?
Yep.
Great, excellent. Yeah, so thanks for hosting this session. I think it went pretty smooth. I'm very impressed. So all our rehearsals really paid off. Just two announcements for the next session. So first of all, if you want to, for anyone in the audience to join the next session, you have to click in the upper left corner. There you see it says schedule session 1 out of 7. If you click on that, a window will open, and then you see the next sessions popping up. So to join the next session at 7 p.m. European time, you have to then click on session 2, memory and navigation, and this will bring you then to the talks for the next session. The second announcement is regarding the Data Blitz presenter. So I think we asked all of you to send us your slides as PDF until 5 p.m. today. So unfortunately, as of now, we only have received about 50% of the talk. So please, if you could send us those slides as soon as possible, because we need to all collate them to one big PDF and then do some other modifications. So please try to send us your slides as soon as possible. Thank you, and then see you all at 7 p.m. European time for the next session hosted by Josh Jacobs. All right.